talk today about a couple of projects on the theme of chemically modified guides and donors for genome editing applications. Of course, when we're doing genome editing, as we've just heard a really nice setup from Dr. Jacoby, we need to think about both RNA and DNA. Cas9 uses RNA guides, a CRISPR RNA and a tracer RNA to cut double-stranded DNA. And so the first part of my talk concerns the use of, or the modification of RNA guides to guide CRISPR to a site of cleavage. And the second part of my talk is the modification of homologous donor DNA molecules for precision editing. And so the assay that we will use a lot in this talk, I'll introduce the first part of it here and the second part of it later, is called the traffic light reporter assay, which can report on both non-homologous end joining, and then also, you'll see later, can report on homologous repair. And so when we create a double-strand break in the DNA and in this construct, that can create a frame shift some fraction of the time, which then allows us to put puts mcherry into frame. And so about a third of the time after an NHEJ event, we see cells fluoresce red. And so we used this assay a lot as a sort of high throughput way to screen RNAs to guide CRISPR cleavage in cells. And in previous work, which is published, and I'm not going to focus on it today, we found that we were able to generate heavily and fully modified CRISPR RNAs pretty well. That's the shorter of the two guides. And you can see we had to pay a little bit more attention to these areas boxed in red, where Cas9 makes direct contacts with phosphates, hydroxyl groups, and nucleobases of the guides. But you can see that the editing efficiencies shown on the top for an unmodified guide, we're able to basically match those using these heavy and even fully modified guides, at least at the concentrations of guides used here. But we found that modifying the tracer RNA, the longer of the two guides, was much harder. And you can see that as we move to increasing degrees of modification, the editing efficiencies drop off dramatically. And again, this is related to Cas9 making direct contacts with phosphates, hydroxyl groups, and bases of the nucleotides shown here. And these really resist chemical modification. They drop potency dramatically. In particular, A15, for example, right here, as soon as we modify A15, we find we lose a lot of potency. And these positions shown here with arrows are the positions that are making these direct contacts. Several of them are in the Cas9 recognition loop. This is this small loop. I don't know if you can see my pointer, but this small loop in the middle, small stem loop, just a two-base stem, is involved in Cas9 determining, recognizing that such a molecule is a guide. So it's perhaps unsurprising that it is sort of resistant to chemical modification. And you can see that as we leaned into trying to modify those positions in several ways, moving in this tracer T6, we left 7 residues that make direct contact to proteinized hydroxyl groups. In T7, those that made direct contact to polar amino acids were left as hydroxyl. Non-polar amino acids were converted to 2' fluoro. In T8, all of those making direct contact were converted to 2' fluoro. And you can see that, again, the activity drops progressively. So there's something quite challenging about these positions. When we increased the concentration to 100 picomoles, we were able to see some editing, although still at lower efficiency with this fully modified tracer T8. So we've needed to move to harder to access modifications. The things that we can buy off the shelf, 2' fluoro, 2' methyl, and so on, just haven't been sufficiently good to allow us to modify tracer to a very significant extent at those 12 challenging bases. One of the modifications that has been very useful is this one, 4' thioRNA. So in this analog, the 2' hydroxyl group is still intact, but the presence of a sulfur in the ring, replacing that ring oxygen with the sulfur, provides significant nuclease stability. So we made the phosphoramidite of that nucleoside and incorporated it into these tracers. And you can see in our previous publication, we were up to tracer 8, T8. And now after several more years of work, our latest and greatest are up to T122. And so there's been a good lot of iteration that I'm kind of going through quite quickly, but we'll submit that shortly, and I'm happy to share more details with you in the meantime if you're interested in the meantime. But I just wanted to highlight that with these modifications in hand, now T121 and 122 are fully modified and maintain full activity at 20 picomoles. Now, if we actually turn up the temperature even a little bit more on our own assays and drop down the concentration another order of magnitude to 2 picomoles, you can see that even here, there's still some work to do. So we lose some activity relative to the unmodified guide. And we think this is probably related to affinity for the Cas9 protein. So full modification still is quite challenging, but we've made huge progress over the past few years. Does this actually matter, I guess, is the next question. And indeed it does. This is a chemically modified guide comparison with unmodified or minimally modified guide RNAs. And we find that when we co-deliver these with Cas9 messenger RNA into cells or in vivo, that we're able to get significantly better editing with the modified guides, as long as they're modified appropriately, than with the minimally modified terminal-only modifications. The differences are subtle still, and you can see this second pattern, C40, T41, doesn't show as much benefit. So we think that modification is important. It hasn't been easy, but we are indeed making progress in modifying these guides. Modified guides can be delivered with Cas9 messenger RNA or Cas9 protein. Most of our work so far in the brain has actually been to deliver these with Cas9 protein. And we've been able to see some genome editing in the mouse brain. It stays pretty close to the site of injection. You can see these brown cells are stained for edited cells that are expressing a fluorescent marker. It's a slightly different reporter, but that's sort of where we are. So Cas9 uses RNA guides to cut double-stranded DNA. That's all good, but in many cases, we don't need to deliver these as 2 separate guides. We can deliver them as a single guide by ligating or joining those 2 ends of CRISPR RNA and tracer RNA. Synthesizing these in one go is pretty challenging. This is 100 bases of modified RNA, which is not an easy synthesis. And so one of the things we've been doing has been to assemble single guide RNAs using chemical ligation. And if you haven't previously read the Tom Brown Group's great paper on this in Nature Communications 2019, I recommend that as well. They, of course, you might expect, used click chemistry, azide alkyne ligations. We explored a different chemistry, and I'll tell you a little bit about that just for a couple of slides, which is the inverse electron demand Diels-Alder ligation between a tetrazine and an appropriate alkene. This goes first through a Diels-Alder, then an inverse electron demand, and then a retro Diels-Alder with loss of nitrogen to produce these dihydropyridazines, and then can be often oxidized to the pyridazine shown here. So it's simple enough, in a sense, to join CRISPR RNA and tracer RNA into single guides using this chemistry. We introduced a tetrazine onto the 3' end of a CRISPR RNA, and we used a norbornene phosphoramidite on the 5' end of tracer. Norbornene is fully compatible with all the conditions of synthesis and deprotection of oligonucleotides, so we didn't have to do anything post-synthetic to those strands to make a norbornene functionalized tracer RNA. And you can see that adding a PEG-8 spacer produced a little bit of extra activity if you compare the red bars, which are linker 2, to the green bars at which are linker 1 at different total dose of RNP into the cells, or different ratios of Cas9 to the single guide. But we wanted to compare these at endogenous sites to the native or standard single guide RNA just made with nucleotides. And you can see that at some loci and at high concentrations, these are picomoles into cells, we were able to show similar performance. But as we dropped down to low concentration, and particularly at some loci like this track locus, we saw that our tetrazine ligated guides didn't perform as well as the standard guides, standard single guides. If we change the ratio of Cas9 to single guide, again, this sort of accentuates the challenge, and particularly at some challenging loci like the track locus or HPRT, we see that there's really a pretty dramatic difference in the editing by standard tetraloop sgRNAs and our tetrazine guides. So we did a little bit of chemistry. We explored other structures of these tetrazine ligated sgRNAs. This is linker 2, which was the best one previously, and we explored several additional structures, extending the base paired stem and changing the linkers between the tetrazine and norbornene groups and the rest of the guide. And you can see that linker 5 was able to match the potency of editing, the efficiency of editing of the tetraloop guide, which is shown in gray, pretty well, even at this challenging locus HPRT. If you recall, previously, there were some really dramatic differences in the editing of HPRT. And indeed, that's borne out again. You can see the red bar at 2.5 picomoles really shows a pretty dramatic loss of activity with linker 2, but this optimized tetrazine linkage does pretty well. So that paper is just about to come out in ACS Chemical Biology, should be online later this week. So in the second part of this talk, I want to talk a little bit about our work on modification of homologous donor DNA molecules to drive precision editing, to add information to the DNA within cells. Of course, when a Cas9 induced double-strand break is formed, it can go through non-homologous end joining and produce indels, or if a donor is given, it can incorporate that donor and produce precise gene editing. And a couple of years ago, a student called Krishna Ganta, who was in Craig Mello's lab at the time, came to my office with an idea. He said, I wonder if we can increase the extent of HDR by increasing the concentration of DNA in the nucleus, and if we can do that by adding a nuclear localization signal to the DNA. So how could we get that onto the DNA? And I thought with him for a few minutes, and we came up with this idea of creating a landing pad of 2'-methyl RNA that could be then serving to base pair to a peptide nucleic acid conjugated peptide. And we would separate these from the DNA donor with a little flexible spacer. And so we went away and made these molecules, and a few weeks later, I got a text from Krishna on my phone. I was down getting some lunch, and he said, where are you? And I said, well, I'm grabbing some lunch. And he said, I need to talk to you. And so I said, okay, give me 5 minutes. I'll be back in my office. Well, he wasn't waiting for me in my office. He was waiting for me at the lift. And so I sort of see the doors open, and it's Krishna's face right outside the door. And he says, it works. It was sort of a very exciting moment. And this is what he saw. So a normal donor produces, Krishna was doing his PhD with Craig Mello in C. elegans, the nematode worm, where HDR was pretty inefficient. So with a normal donor, you can see that we see really very poor editing. Less than 1% of worms were showing the desired genotype. And that meant for a lot of sorting worms and a lot of microinjections to get the desired edit. But in the presence of this 2'-methyl RNA linked by a tag and then annealed with a PNA NLS, about 1/3 of the worms had the correct phenotype. So we thought, this is wonderful. We should do the negative control and show that it's dependent on the presence of the NLS. But when we took the NLS off, to our great surprise, there was no difference. It was still highly active. And that was true across multiple targets. The NLS is actually not important to drive efficient precision editing. So these were the kinds of things that Krishna was then seeing under his microscope. He was incorporating GFP into these worms. And after years of sort of struggling to make this work, now in some injections, the majority of the worms were actually showing the incorporation, which was very exciting. But most of us are not worm people. Most of us, my lab thinks a lot about therapeutics. We're interested in human editing, human somatic cell editing. And so we wondered if 5' modification of the same kind might enhance HDR efficacy in mammalian cells. And to study this, we went back to that traffic light reporter assay that I showed you half of earlier. Now let's look on the left side of this slide, and you can see that if we provide a DNA donor, we can repair a defect in the GFP sequence, which allows us to score green fluorescent cells as having undergone HDR. We found that these 5' modified donors significantly enhance HDR in mammalian cells as well. And interestingly, in this context, 2'-methyl RNA alone or the TEG alone also gave a significant increase in genome editing. The combination of RNA and TEG was very good. And you can see that that increase in precise editing correlates with a decrease in NHEJ imprecise editing events. These donors also show enhanced potency. So at a lower concentration of donor, you can see for a given amount of editing for 20% GFP, let's say, the peak for the unmodified donor, this black curve, we can see that at about 4 or 5-fold lower concentrations of DNA. And again, the amount of the number of imprecisely edited cells decreases as we add this modified donor. This works not only for reporters, it also works for multiple endogenous loci and multiple cell types, including these hard to transfect cells. We see lower extent of editing in these hard to transfect cells, but still a 2 to 4-fold increase in the extent of HDR. So how does this work? Why does 5' modification affect or enhance HDR efficacy if it's not driven by a nuclear localization signal? Well, one of the things that we saw is that 5' modifications suppress the formation of concatemers. So when a DNA duplex is introduced into cells, that DNA begins to assemble through end joining reactions, ligations. The cell seeks to repair double-strand breaks, and this generates the formation of sort of a ladder of concatemers of increasing size. Whereas in the right lane of each pair, you can see that the presence of a TEG group on the 5' end of the DNA blocks the formation of that concatemer. At long time points, maybe that PEG is being cleaved off. And so you do see some concatemerization at long time points, but you can see a pretty dramatic drop. Well, if concatemerization, I should say that concatemer causes two problems. First of all, longer things are harder to traffic into the nucleus. And secondly, more to the point, that would be the incorrect incorporation if a second, if a double copy or triple copy of the insert was incorporated into the genome. But if concatemerization was the predominant factor, then 3' modified DNA donors should be just as effective as 5' modified donors, right? All of this data so far had been on long double-stranded DNA donors derived from PCR using TEG-modified primers. But we then carried out some experiments using single-stranded donors where we could modify either end identically. And we still see the benefit in single-strandedness, particularly in terms of potency. So here's an unmodified donor, the lowest of the blue bars there. And you can see it produces about 12% precision edited cells. When we add RNA TEG on the 5' end, we still see that 5' modifications enhance HDR. If we put 3' modifications, a 3' TEG group, then we see a loss of that enhancement. So 3' blocking modifications are inhibitory. And it's not about nuclease stability because we can put a phosphorothioate modification at the 3' end, but the 3' OH needs to be left accessible. And this presumably indicates that it needs to be accessible for extension, that it's serving to sort of be a substrate for polymerase to further extend it. So far we've talked about HDR improvements in C. elegans and mammalian cells. What about other species? It does work in zebrafish and mouse germline, so I'll show you very quickly a little bit of that data. In zebrafish, this was in collaboration with Nathan Lawson's lab. The unmodified DNA donor or TEG-only donor did not show significant precise edits, but the RNA tag donor did. Really pretty significant improvement. And in mouse transgenesis, these are injecting the zygotes during mouse microinjection of zygotes to create model mice. You can see that this tyrosinase locus is a useful system. These are albino mice where correcting a SNP or a couple of base pair change, actually three bases within the tyrosinase gene allows the restoration of color within the coat of these mice. So while an unmodified donor produces about 10% uniformly dark mice, we can see about 35% uniformly dark, or 38% if you count mosaic mice. So that's both alleles being modified in the zygote. Okay, so that's good for efficacy. We've just heard a great talk from Dr. Jacoby about the precision of editing. And so we used the technique that she mentioned called GuideSeq to explore whether 5' modifications also reduce random donor integration in the genome and therefore increase precision of editing. And so this is a double-stranded DNA donor. It has 2 internal phosphorothioate linkages on all the termini to protect it from nuclease degradation. And we compared it to one with a 5' tag on both strands. And you can see that indeed, we do see a reduction in random donor integration at both on and off-target cleavage events. So that's good news. So this work on the modifications has been just published late last year in eLife in collaboration with Eric Sontheimer and Craig Mello's labs. I want to give you just a little bit of a flavor of what's coming next on that project in just sort of 1 minute. As you can imagine, there are a number of things we can modify. We can modify the backbone of this sort of landing pad RNA group. We can modify the backbone of the primer of our DNA. We can modify the sugars and of course the sequence of this region as well. Do those things make a difference, even if we haven't needed a nuclear localization signal to be attached here? So let me just show you a little bit of data. One of the systems where this might be most exciting would be in the engineering of T cells or other kinds of cell therapies. And so this is work done in a T cell line called Jurkat cells. D1 is an unmodified donor and D2 is modified with PEG only at the 5' end. And you can see that adding phosphorothioates through the 5' end, D3 through D6 with increasing extent of phosphorothioate improves the extent of HDR. If we add phosphorothioate on the donor itself, so the PCR primer essentially, that also further improves HDR. And if we try to make that landing pad out of DNA, that decreases its activity. So 2'-methyl RNA is better than DNA, suggesting that sugar modification is also important. And then in one more piece of data, the sequence and length do matter. Different sequences can show even further increases in HDR potency and efficacy. So I've told you about 2 papers that are out, and in both projects told you about a little bit more work that's not yet out, that's kind of earlier stage work. I hope that that's been interesting, and I'm happy to talk with you about more about both of those projects. Thanks very much for your time, and really need to thank these people too. These have been collaborative projects between my lab, Craig Mello's lab, Eric Sontheimer's, Anastasia Kurova's, and Nathan Lawson's. Thanks very much.